Let me start with a spoiler. So Bax-and-Bach deficiency limits oncogenesis. As we will see, this will be done mostly at the beginning of the oncogenesis, and then we will avoid to get aggressive cancer cells. But also, we know that Bax-and-Bach deficiency confers resistance to anti-tumoral treatments. As we know, anti-cancer therapies, mostly of them, such as chemotherapies or radiotherapies, induce apoptosis in cancer cells. But in the case of Bax-and-Bach deficiency, we won't see this apoptotic pathway. So how it's possible to have this paradoxical function of Bax-and-Bach? Many types of cancer chemotherapy induce cellular stress, leading to induction of apoptosis. As all of us know, Bax-and-Bach are part of the BCL2 family proteins. They are pro-apoptotic proteins with the 4 BH domains, and they are known mostly because they are located at the mitochondria, and they are involved in the mitochondrial pathway of apoptosis. As you know, Bax-and-Bach are in monodimers when they are inactive, but after certain stimulus, they oligomerize, and they can create pores in the mitochondrial outer membrane through which the cytochrome C will be released. However, it's known that Bax-and-Bach is also present in the ER, and they will be involved in the loss of ER integrity and calcium homeostasis, but it's not really clear how it's done. One hypothesis is that Bax-and-Bach interacts with the IP3 receptor and helps to the calcium release from the ER to the mitochondria, because as we know, during mitochondrial pathway apoptosis, there is an overload of calcium. But also in the last years, another hypothesis is that Bax-and-Bach can create pores also in the membrane of the ER and provokes the release of calcium from this store. Some types of cancer therapy induce immunogenic cell death. I went through this pathway in detail, but just to say that some cancer cells, when they are treated with some chemotherapies, such as anthracyclines, and they start to die, they generate some DNA-associated molecular patterns that provokes the activation or the call for the immune system. One of these proteins is calreticulin. So calreticulin is a calcium chaperone located in the ER, and when these cells are stressed, is translocated to the surface membrane, and acts like ITMI signal for the immune system. Then the immune system will recognize and eliminate these cancer cells. In the laboratory of Guido Kremer, it was published also the pathway of calreticulin exposure, and we observed that Bax-and-Bach are involved in this pathway, meaning that in the absence of Bax-and-Bach, there will not be an exposure of calreticulin to the cell surface. So Bax-and-Bach deficiency prevents apoptosis, but also Bax-and-Bach deficiency prevents immunogenic cell death. So that means that it prevents the intrinsic pathway, but also an extrinsic pathway. That means that Bax and Bax deficiency leads then to the cell death resistance. What we have done for the last year, and it was published in November 2021, is that Bax and Bax cells do not tolerate tetraploidization. I'm pretty sure that Mohammed has explained you already about tetraploidization. I'm sorry, I was in the university courses, so I couldn't be present. But I'm sorry if I repeat again, but tetraploidy is considered a precancerous step between diploid healthy cells and aneuploidy cells that are considered the aggressive cancer cells. So normally when the tetraploid cells are freshly formed, they will die by apoptosis due to the tetraploidy checkpoint. However, some of these tetraploid cells will escape to this intrinsic mechanism, and after several events like multipolar mitosis, asymmetric cell divisions, or chromosome loss, they lead to the aneuploid cells. In the past, we have described that tetraploid or hyperploid cells are immunogenic. That means that, in fact, tetraploid cells exposed in the cell surface calreticulin. So, in fact, there is the same calreticulin pathway, exposure pathway, done in anthracycline active stimulus. So we know that tetraploid cells have more calreticulin in the cell surface. That means that the immune system will recognize and eliminate the cells. But some cells escape to this control, and there will be an immunoselection leading to aneuploidy. To be more clear with this part, I will show you some results in which we have diploid and tetraploid cells. We observe that hyperploid tetraploid cells show more calreticulin on the cell surface, and also we observe that when we inject diploid and tetraploid cancer cells into immunocompetent mice, in fact, hyperploid cells develop less tumors, and the tumors appear later, and they grow slower than the diploid counterparts. However, this difference disappears when we inject the cell lines into immunodeficient mice. What we did was also recover tumors, tetraploid tumors that have been grown in immunocompetent and immunodeficient mice, and we observed that the tumors that have been immunoselected into the immunocompetent mice have lost ploidin, and they have lost also calreticulin. We could say that we are in aneuploidy state. And in fact, if we compare the tumor growth of tetraploid cells when they are injected for the first time into immunocompetent mice, and we inject the immunoselected tumors from these mice again into immunocompetent mice, we observe that these tumors that have been immunoselected grow faster and appear earlier. So these are in coherence with the aggressiveness of aneuploid cells. What about Bax and Bax cells? Okay, so we carry out experiments to detect apoptosis in wild-type cells and double KO cells for Bax and Bax. And we exposed these cells to different platinum-based chemotherapeutics, such as cisplatin, oxaliplatin, and carboplatin. And we confirmed that these DKO cells are more resistant than the wild-type counterparts. However, if we use different microtubular inhibitors, such as nocodazole or paclitaxel, which is a chemotherapy derived from taxanes, and also brotenone, we lose this resistance to apoptosis, and in fact we have similar percentage of apoptotic cells than in the wild type. But also we observe that spindle checkpoint inhibitors like RO3380 and that is inhibitor for PLK1, HCD1152 that is an inhibitor of Aurora KB, or M2I1 that is an inhibitor of MAT2. In this case also we observe that the percentage of apoptotic cells is similar in wild type and in the KO cells. So that means that the whole KO cells are susceptible to killing by tetraploidization. But we wanted to know if this is due to the treatment or if it's due to the fact that the cells are tetraploid. To see this difference, what we have to do is a sorting. And from one hand, we will have the diploid cells, and for the other hand, we will have the tetraploid cells. And for that, we have our cells in culture. In the case of the cells that will be used for diploid sorting, they will be cultured for 7 days in drug-free culture medium, and after 1 week, we will sort 100 cells per well. In the case of tetraploid cells, what we do is to seed the cells, and after 24 hours, we treat with nocodazole or cytocalasine D. All the results that I will show you here are with the nocodazole because I didn't want to put all the results that are in the paper, just an example. So we use nocodazole for 48 hours, and then we will get cells that are tetraploid. But at this point, when we remove the nocodazole, a lot of cells will die by apoptosis because, as I told you before, the most part of the tetraploid freshly formed cells, they will die by apoptosis. So if we want to check stable tetraploid cells, what we have to do is to culture them again for 4 days in drug-free culture medium. Like this, the cells that are prone to die by apoptosis will die. The ones that could reverse to diploid will do it. And then what we sort after 1 week will be stable tetraploid cells. And as the same case done for diploid cells, we will sort 100 cells per well. We keep this 96 wells plate into the incubator for 2 weeks more. So that means that this experiment takes 3 weeks. And at the end, we will count in how many wells we have cells, we have colonies. And this is the percentage that we observe here, okay? We carry out this kind of experiment in wild-type cells, in the double KO cells, but also we did this into in the Bax knockout single knockout cells, Bax single knockout cells, and other apoptotic propoptotic proteins, such as NOSA, BEAM, PUMA, and different combination of them. And we observed that in the case of Bax KO cells, we have more tetraploid clonogenicity, and this is coherent with the previous results obtained in the laboratory of Guido Kremer, in which they have observed that the absence of Bax favors the apparition of tetraploid cells. However, what we observe with back KO is that this capacity of clonogenicity for tetraploid cells decrease. But the most important result is obtained with the double KO for backs and back, meaning that these cells are not able to proliferate. To confirm this, we carry out another type of experiment. Instead to see the cells into 96 Wells plate, what we did, it was to recover these cells into bigger Wells plate. I think that it was 6 Wells plate. And we count how many cells there were in the wells for the same time that we have the plates, 96 well plates in the incubator. So at 7 days, 7 days we do the sorting, and then we start to count how many cells there are until 21 days, that is the same data than here. And we observe that at certain moment, the wild type cells were able to start to proliferate. However, the DKO cells were not able to proliferate. We wanted to know if apoptosis was enough, because it must be something different if we have the same level of apoptosis and but we have less cells in proliferation. There should be something else more. So for this reason, we test senescence, and for that we look for P16, P21, and P57 that are markers for senescence. And in fact, we observe that the expression of P16 and P21 was higher in DKO cells treated with nocodazole compared to the the wild type ones. We didn't observe any difference for P57. So that means that tetraploid double KO back cells enter into senescence. Also, we carry out a similar experiment using C12FDG, that is a substrate of beta-galactosidase, by flow cytometry. We did this because in this case, we could compare the whole population, just control and NOCO, and we observed the same than in the Western blots, but also we could select what happened with the diploid and with the more than foreign cells, the tetraploid ones. And we observe, in fact, that mostly all the senescent cells are tetraploid cells. And this effect was higher in DKO cells compared to the wild type ones. And what about calcium fluxes? As I have shown you before, back DKO cells are involved also in the calcium homeostasis. In fact, we can measure two kinds of events. In one hand, we have ER calcium store. The store of calcium in the ER is an indirect measure of cell death because, as I told you, in the mitochondrial apoptotic pathway, there is an overload of calcium coming from the ER to the mitochondria. So if we have less calcium in the ER, that means that they could be more resistant to cell death. And it was what we observed. In fact, the DKO cells at basal level, they have less calcium concentration in the ER. But when we treat the cells with nocodazole, in fact, we lose this small concentration of calcium. I mean, we have more. There is an increase on the calcium ER concentration at the same levels than in wild type. The other measure that we can do is store operated calcium entry, calcium from from outside to the cytosol. And this is an indirect measure of cell proliferation. That means that the cells that have higher store operated calcium entry are able to proliferate more than the other ones. And in fact, what we observe in the decay of cells at basal level is that these cells have more store operating calcium entry. That means that are able to proliferate more. But in fact, when they are treated with nocodazole, this SOCI is decreased at the same levels than in the wild type. In fact, for wild type, there were no difference neither in the ER calcium concentration, neither in the store operated calcium entry. So if we have a role of the ER and the calcium, we wanted to know if double KO that have an overexpression of SERCA, SERCA is the calcium pump in the ER, the main calcium pump, the one is implicated in the entry calcium from the cytosol to the ER. We wanted to test if these cell lines, but also if we have decay cells with BAC at the ER level, just at the ER, restores this observation. So here we have the characterization of double KO SERCA cells and the characterization of double KO BAC at the ER. And what we observed was that both DKO SERCA overexpression and DKO BAC at the ER restores the ER calcium concentration of DKO cells to wild type levels. But also they restores the store operation calcium entry of DKO to wild type levels. And what about senescence? So we repeat the same experiment for senescence, but this time using wild type DKO, DKO with overexpression of SERCA and of BAC, DKO with BAC at the ER. In control and after the treatment, and we observed that mainly DKO cells with BAC at the ER restores the levels of senescence markers such as P16 and P21 of DKO cells to the wild type levels. For P27, in fact, there was no activation neither for wild type and or BAC, DKO BAC at the ER. And the last experiment was to confirm that once we have overexpression of SERCA at the ER or BAC in the reticulum, restores also the clonogenicity capacity of tetraploid cells. And in fact, it was what we observed. Here we check again the 4 cell lines, and we observe that both SERCA cell line and BAC at the ER restores the clonogenicity capacity of tetraploid cells to the wild type levels. What does that mean? So that means that if we have diploid double KO cells and we induce tetraploidization, in fact, these tetraploid double KO cells will enter into senescence, and then we avoid this pathway leading to aneuploidy. So, in fact, what we can say is that BAC and BAC deficiency is not a major driver of oncogenesis because it compromises the proliferative potential of tetraploid cells hence obliterating one of the major paths towards carcinogenesis, as is the tetraploid. And this is consistent with the fact that double KO back adult mice do not develop cancer. We have seen that the double KO mice, most part of them die in the perinatal period. So there is a drastic reduction in birth, but also the most part of them the ones that can birth will die after 48 hours or within the 48 hours. But just a fewer percentage, less than 10% of the animals will reach the adulthood. These animals, we have typical characteristics, but like the membranes, interdigital membranes, or the unperforated vagina. But what I want to say is that they have abnormal brain morphology and they have impaired immune system, even if they don't have cancer. In both cases, there are more number of neuronal cells and more hematopoietic cells. What we have done is in silico oncomine study, in which we have observed that in fact the underexpression of BAX and BAC is present in brain and central nervous system, and also in some leukemias. And this kind of cancer are the most common cancer in children. So what about if there were BAX and BAC deficient tumors? So we can imagine that we have BAC diploid cells. They have a damaged signal. They won't be able to lead tetraploid cells that we know, what is quite good, because these cells would be resistant to apoptosis and they will be unrecognized by the immune system because they are not able to expose calreticulin on the cell surface. But some of these cells can escape, because in any case we have observed that sometimes there are some clones in the double KO tetraploid cells. What can happen with the cells that are able to escape to this damage? So what we propose is that if we have BAC cancer cells, we can treat them with tetraploid-inducing agents, and then we will have senescent cancer cells. And this is the work that we are setting up right now in the laboratory. And I hope that I can explain you maybe in the next time about these results. And I want to thank overall the people that have been working mainly in this project, the three first authors of the paper that have been published in November 2021, that are Jin Deng, Lucia González Gutiérrez, who is at that time was a PhD student in another laboratory here in Valladolid, but right now she has got Margarita Salas postdoctoral fellowship with my laboratory, but she will spend 2 years in the laboratory of Guido Kramer before coming to my lab. And also Gauthier Stoll, who participates for the immunostatistics, for all the statistics. And I want to thank Daniel Barriuso, who is my PhD student right now, and all the projects that is coming up from this one is his PhD thesis. And also Lucia Alvarez Frutos, who is a master student with us right now, and she's helping in the laboratory. And if you have any question.
Well, Laura, it was fantastic, really. very pedagogic. So actually, I have some questions, as you may. So in a certain moment, you were showing that with the platinum, so the CDP, so the cisplatin, etc., the treatment was okay. But when you played with SAC inhibitor and microtubular inhibitor, you had some, so cells are more susceptible to be killed.
Yes.
So for the microtubule, actually, so you tried with NOCO, and yes. So did you try it, for example, vincrescine, which is, you know, the antagonist of locodazole, if I'm not, yes, antagonist. No, vincrescine is the same actually. So did you try it, you know, the antagonist, so vincrescine and paclitaxel, and more especially actually the combination between cisplatin and this spindle inhibitor and microtubule inhibitor.
So I'm taking note because it's a very good idea, and with the pencil, because I didn't try, but it's a really good idea. In fact, we want in the project that we have right now, we use cancer cell lines, and these cancer cell lines are coming from kind of tumor from children, and we want to use not only nocodazole or rotenone that are not specific chemotherapies, but also we want to use paclitaxel, other epithelons, vincrescine, and all of them. And it's a very good idea to use antagonists of them or in combination to add to the studies.
Okay, wonderful. You know why? Because actually I'm also working on a parallel project, and what I found dealing with neuroblastoma cells that when they acquire resistance actually against vincrescine, for example,
yes,
so they show the same resistance when we treat them with paclitaxel. So they are antagonists, you know, vincrescine and paclitaxel. But when we play with some doses of spindle assembly inhibitor, they show so the resistance, we overcome resistance only with vincrescine, but not with paclitaxel. And it was really, I mean,
it's interesting.
I still deal with this. I've been trying to explain why with vincrescine and not with paclitaxel. So so combination, cisplatin, etc. And then the other question, main question actually, what about P53? So you didn't say here, but actually when we do not have P53, we even encourage tetraploidization, and we even facilitate tetraploidization. So what about the P53 statue here in your study?
Yeah, so as you said, P53 is the main protein involved in tetraploid checkpoint. So in the absence of P53, of course, there is more tetraploidization. So I didn't want to have more variables in this project, so I checked the P53 level in the cells, and it was as the same level that in the wild type.
Okay. So
like this, I avoid an effect of P53.
Okay. Then the last question will be, so if we do not have any back, so double cow, as you said, and you stop proliferation and cell undergo senescence. So is there any idea to develop some drugs to inhibit this pathway when we reach like tumor, you know? Is there any idea to develop?
Well, I think that the good idea, but this is really a spoiler of my hypothesis. But okay, I think that the good idea is to use tetraploidizing agents to induce senescence, and in combination with senolytics to eliminate the senescent cells. I think that it would be the best treatment, but of course, maybe we can look for another kind of of treatment.
Absolutely. And now I remember that I note also another question. You know that I have observed that these tetraploid cells, they move faster.
Yeah.
So that by any chance you observe it also when they are double KO back, they or maybe only back or only back. Did they have any change in migration, moving?
No, but you are really doing a spoiler of my project. So what we want to do is to inject these cells into the mice, in immunocompetent and immunodeficient mice, to see if there is any difference that we expect that in fact they are not recognized, because normally they should happen have reticular surface and they won't be recognized, and check if they are also more aggressive, meaning that they are able to induce metastasis more than the wild-type cells.
Okay, good. Thank you again, Laura.
Thank you. I think that we should collaborate.
Absolutely, no, absolutely. We must.
Yes. Okay. So stop to share, or.
Yeah, maybe Kamel, I assume he has some question, and
also you know, I have no further question, but you already you know, asked a very interesting question, and you are very close to the field. I have just a naive question about what's about human, you know, how involved with backs in human cancer.
Yeah. So in fact, it has not been described knockout of the two alleles of backs and back. This is true, but I can say that we have observed deficiency of both proteins in some cancer cells, human cancer cells.
Okay.
So it could be a deficiency, but not completely knockout.
I understand.
Good.
Thank you.
Any other question in the audience? So if not, again, Laura, merci beaucoup. Muchas gracias.